Hi guys, Kobe here for Science Sapien, and today we're going to talk about biochemistry. I'm just going to do a voiceover on this presentation. This is an overview of biochemistry. It's great for review for a test or the SAT, but keep in mind there's a lot in biochemistry, and we're only going to talk about some of it and kind of overview a lot of it. This is part one of two of the presentation on biochemistry, so if you want to see the second part, head over to my YouTube channel, Science Sapien, and you can see the second part. Okay, so biochemistry is the study of the molecules of life, and they basically make up all of biology, and because of that, they're really important to study. So we're going to talk about a bunch of chemical interactions, how chemicals and atoms build up and create what we know as life. All right, so before we get into any biology, we have to talk about purely chemistry. And subatomic particles are the particles that make up atom, and they have three main parts. They have a proton, a neutron, and an electron. A proton has a relative mass of 1 and is positively charged, and it's in the nucleus, in the center of the atom. And the neutron is also relatively large. It has a mass of 1, but it doesn't have any charge. And it sits in the nucleus with the proton and stabilizes larger molecules. And then electrons have a mass that's really, really small, but they have a negative charge. So a charge equal to the proton, but negative. And they're in a shell on the outside, and they determine a lot of the features and the characteristics of atoms. Okay, so atoms in the elemental state always have a neutral charge, meaning they have the same number of protons and electrons. And the number of protons in an atom determines what kind of atom it is. Okay, the electron configuration, like I said, determines how the atom will react, what kind of characteristics, personality that atom has. And then finally, electrons are really lazy. They're always going to be in the lowest open level of possible, which is called the ground state. And electrons do this because they're basically lazy. They want to have the least amount of energy as possible, so they go into the ground state. And this is important to understand because these subatomic particles interact when atoms bond. Okay, and then an excited state is when these atoms get energy from some source, maybe the sun, maybe from another molecule, and that causes them to jump up to a higher level, which can cause all sorts of things like bonding, or they're really important in processes like photosynthesis and cellular respiration, where cells use molecules to create energy for themselves. Oops. Sorry. All right, so isotopes are atoms of an element that vary in the number of neutrons. So, for example, hydrogen has one proton. That's what makes it hydrogen, has one proton. But it can have more than one neutron, and that will change just kind of the shape and stuff. But it's still hydrogen. It'll become heavier, but its charge will stay the same, and it's still the same molecule. Radiation is a process through which molecules actually lose energy, and they lose this through losing protons. So something like uranium which is radioactive, is a molecule that's radioactive. It will lose protons, lose energy, and become other molecules. And this is really important because it allows us to date things. So a half-life is the time it takes for half of an isotope to decay, which basically means if we start with, let's say, 100% carbon-14, which is just an isotope of carbon, we know that over time carbon-14 decays at a constant rate of 5,730 years. So if I have an organism and it has, let's say, 100% carbon-14, and I dig it up 5,730 years later, it now has 50% of the original carbon. So let's say I'm looking at woolly mammoths, and I know how much carbon a live woolly mammoth would have because I know the atmosphere. Let's say I dig it up and it has like a quarter of the carbon-14 I expect. That allows us to date the mammoth because we know how much carbon-14 is left. We know what percent that is of the original, and we can use that to date the mammoth. So carbon-14 dating is really important in biology and in dating organisms, dating fossils. And then in medicine, isotopes are really important too. They can be used as tracers. So if you want to see a certain type of cell in the body or something like that, you can inject a tracer and follow that through imaging software and imaging tools, and that allows you to track molecules and diseases in humans and other species. All right. So bonding is when we have two molecules that interact with each other. And this is really important because very little in biology is just one atom, just one element. A lot of stuff is multiple elements. Glucose, for example, is C6H12O6. It has 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. And they bond in a very specific way. So because biology uses these big complex molecules, we have to understand how littler ones combine to make these molecules. Okay, and there's two main types of bonding. There's ionic bonding, and what happens here is one molecule loses an electron, the other one gains it, and because of that, they're basically like magnets. They have one of them's positively charged, one of them's negatively charged. That's what we're seeing here, right? So this one here is going to lose an electron and give it to this atom here. Because this atom now has more electrons than protons, right? It has 2 electrons and 1 proton. It's got a negative charge, a charge of -1. Whereas this has 1 proton and no electrons, so it's +1, positively charged 1. So they basically act like a giant magnet to each other and they stick together, and that's called ionic bonding. Okay? An anion is negatively charged and a cation is positively charged, right? The T in cat looks like a plus. And then the other type of bonding we have is called covalent bonding. It's created when molecules share. So when they're not greedy and they don't steal, they share, right? So here we see that both of these have 1 electron, but they want 2, okay? And because they both want 2 and they each have 1, they can actually share both of them. So they're both happy, and this is called covalent bonding, okay? And covalent bonding is more common. Ionic bonding leads to stuff like salt, but covalent bonding is used a lot in biology. All right, so those are the two types of bonding and the ways that atoms interact. All right. A molecule is more than one atom. And then when atoms create more than one molecule, they also have different types of interactions. Okay, so one is polar to polar. So an atom can be slightly charged. So as we see with this water here, we've got this slight negative charge on the oxygen and a slight positive charge right here on the hydrogen. And what that does basically is that each of these water molecules, each of these H2Os have a slight positive end, right, and a slight negative end. And every one has the same thing, all right? That's slightly positive. Right here is slightly negative, slightly positive, slightly negative. And because of that, we see that these atoms want to stick to each other, just like magnets again. So water is... it's actually sticky. It sticks to each other. It makes these hydrogen bonds, which are polar interactions, because both, all the waters have a positive end and a negative end. They want to stick together. They line up so that all the positives attach to all the negatives. Okay, so that's polar to polar interactions. A specific example of this is hydrogen bonding, right? We see that in water, and it also holds DNA together. So right here, we're seeing that this chemistry is already applying to biology. So the 2 strands of DNA, which you guys, if this is your first week or 2 of biology, or you're just starting biology, you'll talk about it later, but DNA is stuck together with hydrogen bonds, with polar-to-polar attraction. And we also have non-polar bonds. They're not as strong, and they're also called van der Waals forces. And basically what it is that if I have a bunch of molecules, so let's say, here's a molecule, here's a molecule, here's a molecule. They're all non-polar, meaning they don't have slight positive and slight negative charges. They're just kind of the same all the way around. But what happens is because the electrons, like we were talking about earlier, float around the nucleus, they're kind of free to go wherever they want. Once in a while, all the electrons are going to add up, just by chance on one side. So that's going to make that side negative, and the other side will be what? It'll be positive, right? And if this lines up perfectly on several atoms, so this atom here also by chance has negative on one side and positive on the next. Let's say this one does too. Right, just like that, all positive on the right, all negatives on the left. That will cause a weak attraction. And things like geckos actually use nonpolar van der Waals attractions to stick to stuff like walls. It's kind of like static electricity at the atomic level. But these are pretty weak, and they don't last as long as polar-to-polar attractions. DNA is really tough. It will stay together for thousands of years, possibly, but something like a van der Waals force, maybe a few seconds, maybe a minute if you're lucky. They're much shorter. All right. So we also have something called hydrophobic and hydrophilic. Hydro meaning water. Hydro meaning water, right? And phobic is scared of, right? You can be, a phobic or have a phobia that's being scared of. And philic means lover. So molecules can either hate water or love water. And what does that mean? Well, water, like I said earlier, is polar. It has a charge. It has a slight positive and slight negative. So hydrophobic molecules, water-hating molecules, are nonpolar, meaning they don't dissolve in water and they don't like water. So they don't have a charge, so they don't mix well with water. So something like an oil is hydrophobic. If you put it in water, it's going to separate itself, right? That's because it doesn't like the water. It doesn't dissolve in the water. And that's really important in biology because it allows cells to separate themselves from the outside world. Then hydrophilic is water-loving, so these are other polar molecules like salt, right? You put salt in water, it disappears, it dissolves, and that's because it loves the water. It is also polar, so it sticks with the water, and they blend together really nicely. And this is obviously important for biology. Your blood is basically water, but it has a whole bunch of stuff dissolved in it. Those are all hydrophilic molecules. Okay, so just kind of an experiment. Why do sodas lose their bubbles? Sodas are more or less water with some CO2 in it. And CO2 is not polar. It's non-polar, meaning it's hydrophobic. It's scared of the water. It hates the water. And because of that, what it wants to do is escape. And the best way to escape is up. So soda loses their bubbles if you leave it open for a while because the carbon doesn't want to be in the water. So what it does is it rushes to the surface and escapes the water, escapes into the atmosphere. So if you leave it open for a long time, these bubbles are going to run away from the water because they don't like it, and your soda will go flat. So, don't leave your sodas open. Thanks for watching part 1 on biochemistry. Head over to my YouTube channel at youtube.com/thesciencesapiens to check out the second half of this video. And as always, keep questioning.